Welcome back, Bio 2 students. I hope everyone's doing well this morning, this evening, or whenever you're watching this. Today, we're going to talk about the phylum Mollusca and the phylum Annelida, and we're going to get a little bit into the circulatory system and a little bit of comparative anatomy and physiology on these animals. So let's start off with just a quick reminder of things we've done before. Here's our cladogram of animals. There's a couple of different cladograms on this depending on whether you use structure or DNA. I'm going to stick with this one just to make it nice and simple. So we're going to go through that. And once again, this is a list of several evolutionary events. And we kind of went through this already, talking about tissue levels of organization and what the blastopore becomes and so forth. The Lophophorate phyla, a little bit about the trochophore larva maybe when we talked about that larval stage, and ecdysis, shedding skin. So we'll go through those again, but let's get into the phylum Mollusca today. The phylum Mollusca includes things like snails and clams and that sort of thing. Octopus are in this group, squid, cuttlefish, so several things in the phylum Mollusca. Excuse me. So most of them are marine, but there are some that are freshwater and there are some that are terrestrial, like if you think about land snails. Snails are a good one. Most of them, but not all, are protected by a shell, a covering. If you think of a snail, once again, calcium carbonate shell. Also, as I mentioned, the amount of content as we move forward in Bio 2 increases dramatically. There's more information to learn, but once again, also, I think people find that they do better because in part, we get near the end, for one. We get past the midway point, and so people sort of rally to finish off. But also a lot of these things are more familiar to you. So like when we talk about a snail or a clam or an octopus, you have some idea at least of what those kind of animals look like compared to, say, when we did the protozoans, you know, like the alveolata and those kind of groups. Okay, most of the mollusca, nearly all of them, have a unique feeding structure called a radula, which is like a file they use to scrape things. And most of them also have an open circulatory system, which I'm going to explain a little bit right now. And then we'll go more into detail on this later on. But animals that have an open circulatory system include things like the arthropods and most of the mollusca. And because they have an open circulatory system, they don't really have blood, but instead what they have is sort of a combination of blood and lymphatic tissue, and we call that hemolymph. And I'll show you a picture of kind of why that is in a minute. And then in animals like other mollusca, some of them, such as squid and the octopus, different species of octopods, if you will, have closed circulatory systems, nematodes, annelids, and of course vertebrates. And they have a separate tissue. They actually have blood, and then they have interstitial fluid. And I'll explain that. I think the best way to see that is in this picture here. So what happens in something with an open circulatory system is they have a structure like a heart that is used to pump fluid around. And what happens is the heart pumps and it pushes the fluid, but rather than it going to a particular target site, it travels somewhat in a particular direction. But then if these are the cells of the body, like let's say these are cells that make up a muscle or the liver or the kidney, the fluid that comes out of the heart sort of just sloshes around in this space. And that's why we call that an open circulatory system. And then some of that fluid then will flow back into the heart. So again, that's an open circulatory system, and it's called open because the fluid does not stay necessarily inside blood vessels like in ours. So we have a closed system, a closed circulatory system. So this is us right here, and some of the mollusca and some of the annelid worms as well. So what happens in us, in a closed circulatory system, you also have a heart that pumps, but that blood travels through it shouldn't come out, but it travels through the blood vessels, and then it gets to smaller blood vessels, and then for the most part, the cells stay inside this network of vessels. And things like water and carbon dioxide and sugar, things that are fairly small, those will come out of these capillaries to surrounding cells. And so because the blood cell part generally stays inside the circulatory system, we call that a closed circulatory system, and that's the difference between open versus closed. And again, we'll go into more detail on how the heart operates and works in that, but that gives us a start. And the other thing I should mention is that an open circulatory system is generally found in animals that are small, because the heart can pump this fluid and it can slosh around in this area, and it doesn't have to go a very particular direction to get all the cells to get fluid. In a small body design, it's sloshing around in the middle there, works out fine. But as an animal gets bigger, so an animal that is faster moving, larger in size, or a combination of those two, such as you find in mammals and birds and some earthworms, also you need a closed circulatory system because you need to be able to regulate where the blood goes or where it's traveling to. So when we talk about the mollusca, we always start with what we call the ham, and the ham is what we refer to as the hypothetical ancestral mollusk. So what that means is the ham really doesn't exist, but it's thought that this is probably what the original mollusca looked like. So it has these basic body parts. It has a foot that you can see there, a muscular foot. It has a mantle, which is a thin tissue right here that covers the organs, and the mantle secretes the shell that's on the outside of the animal. Then you have what's called the visceral mass, which is the space in the middle where all the organs are at, and all the tissue and things of that sort, all the like the kidney and the heart and the stomach and all those, and those are usually underneath the mantle. And then wherever the mouth is at, there is this structure that's attached to muscles, and that's the radula. And depending on the animal, they use the radula in different ways, but they push the radula out often and scrape algae off rocks and things of that sort. So we have several different phyla that we'll go through. One phyla, sorry. So the phylum Mollusca has several different classes that we'll go through. We'll go through each one of these: the Monoplacaforans, Polyplacophora, Gastropoda, Scaphopoda, Bivalvia, and the Cephalopoda. And these are the different common names of the groups that we'll go through here. You don't have to worry about remembering Neoplinia there, because there's only one of those, and so that'll be okay. So the first class is the Monoplacaphora, and the Monoplacaphora are probably most like the ham, or the hypothetical ancestral mollusk. They are evolutionary, kind of an old design. They were thought to be extinct, and I believe they found them in 1952. They were sort of rediscovered being alive. We just have this picture of one. We don't have any in lab. They have a single shell. They have a segmented body, which is kind of unusual for most mollusks. They're a deep marine organism. They have a reduced head, so reduced levels of cephalization. So it's kind of an evolutionarily old trait. They do have a foot, though, and they have that radula. And again, the radula is used for feeding, and the radula is kind of a unique mollusk characteristic. Anything that has a radula is pretty much in that group. Then we have the Polyplacophora. A buddy of mine at Cal State Fullerton is the world's expert on polyplacophorans, also called chitons. They're all marine, and they have these 8 overlapping shells. If you count them, 1, 2, 3, 4, 5, 6, 7, 8. 8 overlapping shells in the Polyplacophora. They have a foot that's used for locomotion. They have a reduced head also, and they have a radula. And on tide pool trips, you can find these animals on rocks where they hold on to the rock with that muscular foot, and they use that radula to scrape food and algae off of the rocks. Okay, then we have the Gastropoda. This is probably the group you're going to be most familiar with, maybe, I would think. And those are the snails and the slugs. There's a couple really interesting things that happen when you look at a Gastropoda. Not all of them are this way, but many are. But they come in both marine, freshwater, and terrestrial types. So you can find land snails and freshwater snails and marine snails, and then slugs that are in those same categories as well. They have an unusual body that results from a process called torsion. So what happens in torsion, which we need to be careful to make sure we distinguish that from coiling. So there's 2 processes here. One's called torsion and one's called coiling. So we'll talk about both of those. What happens in torsion is if you had, say, your ham or your hypothetical ancestral mollusk, in torsion, the body is twisted like that. It's turned from the top. If you were to grab the snail, say by the top, and twist it, you would get a turn like that. And what that ends up resulting in is if you had the mouth here, and then you would expect the anus to come out the other end. Well, because of the torsion, the anus actually sits and drops right over, kind of close to the top of the head. And there's all kinds of interesting theories on why the evolution of that might happen. But whether they're true or not, I don't know. It's hard to say. But bottom line is it does happen, because during development you do have this turn like that, so that you end up with torsion where the anus ends up sitting sort of right above the head. Okay? Torsion. That is different from coiling. Coiling is a turning that's happening like this. If you were to grab the animal from the back and twist it like that. So torsion is like that. Coiling is like this, or like this. So coiling comes in 2 forms. It can be dextral or what they call sinistral. Dextral is a turn to the right. So this is a right spin or turn on the shell. Okay. And then sinistral is turning to the left. So if you're turning the animal this way, that is dextral. If you're turning the animal that way, that's sinistral. So it's a right hand coil or a left hand coil. They also have a foot for locomotion, and they have a radula as well. The next group is a Scaphopoda, and the Scaphopoda are benthic marine organisms. Benthic means they're on the bottom. That can be deep water or shallow water, but they're buried in the sand on the bottom, and they are a type of filter feeder. Filter feeders are things that capture food that is floating around in the water. And so what happens is they have this foot that they use to bury in the sand, and then they also have a radula used to move food to the gizzard, but water and nutrients come down like this essentially and then get pushed back up. The common name for these animals, they call them tusk shells. So in lab, what you would see is this animal we have just the shell of, it'll be like this, and it's like a long tooth or a tusk, and it's open on both sides. One side's bigger than the other. Then we have the bivalvia next, and bivalvia are the clams, and also called mussels. Some of them are called mussels, different species, but they're called bivalvia. They have 2 shells. They're all found in water, so none found on land. They're marine and freshwater, and they have 2 shells, flattened shells with 2 valves, one on each side. And one of the things you got to be able to do is look for this swelling part. That's called the umbo. Actually, I don't know if I see umbo written there. So I see hinge area. Anyway, that big swelling is called the umbo. And the umbo distinguishes where the shells sort of connect together. And it also allows you to know right, left, front, and back. So what happens in a clam, which is a bilateral organism, if I were to draw a line like this part is the head. And then this is posterior, or the back of the animal. And so what you're seeing right here is, if you imagine the head that way and the feet this way, if you were a human laying this way, this shell that you see in the picture is the right shell, and the one that's missing is the left shell, because this is the head, this is the right side of the body there, and the left side has been removed. They have a reduced head, and they are filter feeders. I just said head here, but mainly I'm distinguishing, there's the mouth, and that's a head-like region, and that way you have an idea that that's the head or anterior part of the clam, and you need to know that to know right and left. They're filter feeders. They have siphons, so what they do is they have water in the in current siphon sucking water in. It goes over and gets captured, and then it goes out like that. And they have no radula. They're the only bivalves to not have a radula. They have the other characteristics of it though, but they are filter feeders, and as a filter feeder you probably don't need a radula for the most part. The next group is the Cephalopoda. In the class Cephalopoda, the term cephala means head. We've seen that before. Poda means foot. So these are the head foot animals. They're all marine. They have a head surrounded by tentacles. They have a shell that's either external, like you see in the chambered nautilus, or they have an internal shell, or it's absent altogether. They have a mouth with a radula on it that they use for hunting, and they have a locomotion by using either their arms, depending on what they're doing, or by a siphon. So there's a siphon where they have where the mantle's folded, and it forms like a funnel, and they can push water out of that rapidly and move very quickly. And they have a closed circulatory system. So again, that means that the blood portion stays within the blood vessels under normal circumstances, and what comes in and out of the animal instead is fluid like water and things of that sort in those capillaries, unlike an open circulatory system where that stuff sort of sloshes around and mixes. Okay. Next we have the we have the phylum Annelida, which includes in the Annelida the giant Gippsland earthworm in Australia, which on average gets 6 feet. Everything's big and scary in Australia. They found some as big as 15 feet. It's found in a very particular part of Australia, and only like on certain farms, it seems. So it's kind of it's an endemic species to very particular parts of Australia. They have repeated body segments and specialized segments, and those segments are then connected. And when we do the earthworm dissection, you can see that in more detail. Several different classes. So we'll go through the Eligiochaeta, which are our earthworms, the polychaete, which are marine worms, and then the leeches. So first, we'll start with the Eligiochaeta. The Eligiochaeta are our earthworms. This is my typical earthworm here. This is the head, and you can tell where the head is because it is on the shorter side of where this structure is, which is called the clitellum. And the clitellum is a structure used by the earthworm for reproduction. It secretes like a slime around it. And on the shorter side, you have the head. They have no parapodia. I can't really show you something it doesn't have right now. I'll show you that in a minute though. Parapodia are like little feet, and the Evolutionary do not have them, but I'll show you those in a second. They also have, which is not shown very well, not shown probably at all in this picture, they have what are called setae. Setae are little tiny hairs that stick out of the earthworm or worms. And so if you imagine there's little tiny hairs that would stick out of this. They're very hard to see. You'd have to see them probably under a microscope. But if you're ever digging in the dirt and you have an earthworm and you're trying to pull it out, the reason it holds onto the dirt so well is those little setae are able to grab the dirt. And if you were a normal kid and you pulled the earthworm out, the earthworm breaks. But what we're finding is kids don't dig earthworms up anymore. I'm always sort of saddened by how many students will admit that they've dug up an earthworm. And maybe you're just lying about it. But I find that more and more kids, or more and more students, I should say, grew up as kids in which they didn't dig in the dirt and they don't know what I'm talking about. But if you dig up an earthworm and you get half of it up and you try to pull it, not a video game earthworm. So I guess I should qualify that. I'm talking about like the real ones outside. So not in Animal Crossing or one of these games, just to show you I'm up to speed. So I've got two kids. One of them probably doesn't know about digging up earthworms like this, but I know I have another kid that does. So the time I'm making this video in April 2020, the big rage right now is not just the COVID-19, but also the game Animal Crossing, which is all the rage among the kids and maybe young adults too, maybe old adults, I don't know. Okay, then the last group then is not the last group, but the next group is the polychaete. And the polychaete have well-developed heads. They have parapodia, and again the parapodia are the little feet. You can see that pretty well right here. They're like little paddles. So if I had my earthworm like this, or not my earthworm, but my polychaete worm, they have these little paddles on the side of the body like that. And those little paddles are the parapodia. And then often sticking off the parapodia, they also have these little setae, which again are the little hairs. Some of them are tube-dwelling worms and some are free-living. Some of them have jaws on them and they can actually bite you and pack a pretty mean punch if you mess with them. Some of them live in these tubes like this. So when we go on our field trips, you'll see these worms that you'll see just these tubes, and inside that tube the worm comes out when the water's high. Now when we go, you won't see this part because it's only when the water has come in, but this is what the tube worm looks like when it comes out to capture its food. And then the last group is the hereedenia, and the hereedenia are the leeches, also called the leeches. And when you think about leeches, people usually think about the parasite leeches that suck your blood, which they do, but some of them are predators and others are scavengers. So there's a couple of different, many different species of hereedenia, and some of them suck blood, but not all of them do. They usually have a flattened body. They have a reduced number of segments, and they have a reduced coelom. Remember, coelom is that body cavity that we talked about. They have setae that are absent, so they don't actually have the little hairs on them or the parapodia. They have suckers at both ends. You can see the sucker right there where it's allowing it to hold on. And as I said, they come in different types. Okay, so we're going to stop there. That covers a little bit of the circulatory system. It covers the phylum Mollusca and also the phylum Annelida. And we'll stop there and keep moving forward. Hope everyone's having a good day,